My name is Brad, and I'm here from Bioinformatics Corps at Harvard School of Public Health, and we'll talk for a few minutes about work on community-developed variant calling pipelines. So these are built to address a few challenges we encountered in trying to make our tools available to everyone else and build a community around them. And those sort of challenges are that these pipelines are super complex and rapidly changing. So this is just the version changes of one tool, GATK, since I've been working on this pipeline. So it's been through 4 different versions of best practices where, I don't know how many different releases where the options change and you have to mess with things and things get deprecated and changed. And now they even have a new caller that you have to assess. And so, I mean, just for one tool, every 6 months, literally, if you don't change your pipeline, it's going to be outdated and use old things. So we're in a space right now with this where things are changing rapidly and it's hard to keep up. And in addition to just one tool, these all have a large number of specialized dependencies. This is another tool, a huge sheet from Stanford Bioinformatics to show that there's a few of these. But they list all their dependencies out, and there's a ton of different dependencies, and all of these sort of are rapidly changing over time. And it makes a big difference what method you use. If you compare variant callers, you're going to get different answers. And this is a nice tool called GCAT that does comparisons of different alignment algorithms, variant calling methods, shows overlaps, comparison to reference standards. It's a really nice tool to compare. This is just an example to show, like, if you use 3 different aligners, the same variant caller, you're going to get different answers. And so there's a big question of which one should you be using, and that's hard to assess currently. And the other problem is that when you're trying to share these things and parallelize them, there's a whole boatload of clusters that are all almost identical. So, you know, some people are using LSF, some people are using SGE, some people are using Slurm, some people on Amazon, some people Torque. There's a mess of these, you'd like to be able to share with all those people and have a generalized solution. And so I think the approach that will help with this is community. So I guess it's no big thing to say that here, but in this space, it's definitely, there's a lot of variant calling pipelines, each of which is used at a different place and is hard to generalize to a new place. And so my goal with this was to try and develop something that is actually used in multiple places and is contributed by a lot of people. And so it's a great book if you're into community. Up here, Block is a really nice, he's a really good author and describes a lot about how to build and maintain communities and things involved with that. Doesn't have anything in my talk, but it's a good book, and it has the word community on it, so that's awesome. So this is the high-level overview of what we're doing. So the pipeline is called VC Bio NextGen, and it's really an experiment in how easy you can make it to go from nothing to a full, really good variant calling pipeline. And so if you're not familiar with how this works, you start with sequencing samples. So it's human resequencing samples come off an Illumina sequencer and a configuration file. And you feed it into this giant arrow here, which handles running best practice pipelines, integrates a ton of different tools which don't easily work together, and it scales and provides resiliency to failure. And out the other end, you get variations. That's the raw material, so SNPs, indels, and structural variants. And you also get quality metrics associated with that. So it's easy to produce something, and it's hard to assess how well that something is doing. And so it drives me crazy when I don't know how well something's doing. So there's a lot of emphasis on the quality, alignment quality, variant calling quality, coverage quality. And in addition to that, you want to produce something that people can analyze and work with. So there's a big emphasis on trying to produce annotated variants that you can query directly and visualize and work with. So the sort of 6 main areas that I'll talk about today, I got 6, that's a lot, is the sort of goals of the project. The first is to make the variant calling quantifiable so you know what you're doing. The second is to make it analyzable so it's easy to feed into other tools and analyze. Scalable so you can run this on big data sets. Reproducible so you know what you did and can explain what you did and do it again if you need to do it. Community developed, so there's multiple people working on it and it's easy for other groups to pick up, and accessible so it can be used by both biologists and non-scientists. And so I think one of the critical things is being able to quantify the quality of your calls and do well. So the Genome in a Bottle project is a great initiative from NIST, the National Institute of Standards and Technology, that is developing a reference genome for NA12878, which is a widely used human sample. And what it gives you is a real-life reference you can then compare calls against. And so what we've done is use that as a basis to be able to easily compare the calls and assess the quality. And so the details of this aren't super important. What is important is that you're looking at 3 different variant callers, having your freebase, GATK, and GATK applicative caller, and you can tell by SNPs and indels which one's doing the best on each. You can look at the discordant variants and assess what type of regions they're in. So this one, the low coverage regions, they differ a lot in how they call in low coverage regions. They differ a lot in how they do in indels. And you can make assessments based on this. And this is all automated, so when GATK, I literally do this, and then as I'm finishing comparing it, another GATK version comes out. So you can do another analysis and compare and see how that's doing. And it makes a huge difference version to version how they're doing, which one's best. And so if you're interested in this sort of thing, I have a blog post that is linked at the bottom that describes in detail these sort of comparisons and how we're doing. And I think in addition to the things that we know how to assess, there's also things that we like to pretend that we're missing, that we like to pretend don't exist, and those are known unknowns, I guess. And the 2 things we're really making an effort to work on right now are coverage. You get a nice report of your variants and what you call, but what you don't get a coverage of is what you might have missed. And so key genes and pathways you're interested in, did I completely miss those in my variant calling or my pull down? And so there's no way I could have seen them even if there was a variant there. And structural stuff has a huge impact on, especially cancer. I think is a place where we're still really struggling to call these things well. And so these are areas where we're focusing as well, in addition to the SNP calling. So after you're relatively sure, have some probability of sureness that you have good calls, the next sort of step is to be able to do analysis on those calls. And so we're focused sort of in 2 areas there. One is making it easy to take these calls and query them and work with them. So we're using a tool from Aaron Quinlan called Gemini, which is a really nice way to take a variant calling pipeline, a set of variant calls, stick them into a SQLite database, and associate them with a lot of other associated metadata. So you get dbSNP, you get encode, you get pathways. Everything you want to associate with it is all jammed into a SQLite database table, and so you can query it straight away. So it scales up to large populations, so you put everything in a big VCF. You don't have to deal with that anymore. It's all in SQL, and you pull it straight out. It has a nice SQL interface, it has a nice Python interface. It's a great tool. So we export directly to that, give people that, and that's been really productive for us. We have lots of biologists who have some SQL knowledge picking that up directly and working with it. And the other avenue we're doing is feeding into visualization tools. This is a tool called 08 that we're developing, is to look at different quality metrics associated with variants. You can compare across multiple calls and subset those based on depth, all the sort of quality metrics that you get in a VCF file. So it's a web-based way to take those, export them. This exports to genome space, exports to Galaxy, back and forth. So you can take your tools, put them into this, your variants, and put them into this, subset them, put them in genome space, take them to an analysis tool, and work on them from there. So we think this is really valuable to try and get the pipeline, not just static outputs, but also stuff you can analyze directly in other tools. And because we're getting more and more huge data sets, so everyone talks about that today. I didn't have a slide about it because we got huge data sets, but we have projects coming with 1,500 whole genomes, and it's scary to think about how to do that. So there's a big set of work on trying to parallelize these tools, and so we build on top of the IPython framework, which provides a parallel infrastructure, and then use that to build up these heterogeneous clusters. So the difficult thing with these sort of pipelines is each tool parallelizes better in a different sort of way. So stuff like alignment parallelizes well on multi-core machines, so you can keep everything off disk, not hit I/O penalties, and just pipe everything straight through. So it works well that way. Other stuff you can split by genomic regions and parallelize over the whole cluster and get more scaling that way. And some stuff isn't even parallelized yet, so you're just sticking it in one per sample. So we have a way to manage these sort of heterogeneous clusters on top of all those different architectures that I talked about before, and supporting those. And the thing that we, as we're trying to scale this up, is that we had a naive approach for just splitting by chromosome. The problem you run into is that chromosome 1 is really big, chromosome 22 is really small, so you process all your small chromosomes and you're sitting there for 3 days processing chromosome 1. So we've been working on ways to identify regions we can split in and make it more homogeneous, so you're not stuck, didn't finish 99% of the processing and you're stuck waiting for that 1% to finish. So we're doing this based on coverage, trying to select regions across all the samples that are missing coverage, and have some nice stuff using bed tools and high bed tools under the covers to check those regions and select them. And so I guess that's all the science in terms of doing analysis, being able to scale, and then the next sort of thing you want to be able to do is reproduce and let people know what you did. And so we sort of take two different approaches to this, I guess 3 different approaches really I'm talking about. The first is having a configuration that you can revision control and keep up to date and know what you've been changing over time. And so we do this by being able to express your intentions at a high level in configuration file. The second is being able to handle distributed logging in a way that you can actually see everything that's happening, all the problems that are coming out, all the good things that are coming out, all the command lines, and log those well, and then trying to add higher level provenance tracking on top of that. So this is the configuration file. So if you like YAML, this is a nice slide of YAML for you to look at. And basically it's just a high level sort of domain specific language to describe what you want to do. You know, I want to use BW as an aligner, I want to recalibrate with GTK, I want to call with GTK freebase and haplotype caller, and what type of sample it is. But the great thing I think that's sort of underappreciated is that it's just a text file, so you stick it in revision control along with your scripts, and you're automatically have a backup of everything you've done over time. You see how you changed it, you see if you've made a mistake over time, you see why your calling changed, and you synchronize that with your outputs by date, and you've got an easy way to be able to reproduce and say what you did before. So it's a nice sort of lightweight way to get reproducibility with version control that everyone's already using. And we'd like to build on top of this to make it more queryable. So one of the things we are looking into is moving beyond sort of this really nice logging structure. So we have good logging. We use 0MQ, so you can do things, everything distributed, jam it into 0MQ. It takes care of handling all the distributed stuff. You actually get all your logs back out. You get a big messy log file, but you have everything there. So if you've ever tried to debug on SGE or something where it's jamming your logs all over the place, it's kind of nice to have it all in one place. So that's a cool feature, but we're hoping to move beyond that into stick things into a queryable database, so you can, if you have problem A, you don't have to dig through 27 log files to figure out how A changed over time. You can actually just do a query and find that out. So there's a couple tools we're evaluating for that. One is called BioLite, which is from Casey Dunn's lab, and that's sort of a triple store kind of approach to look at how things change over time. And then there's a tool called Herbados, which came out of George Church's PGP project. And that's a tool specifically for running pipelines and evaluating provenance over time. So those are both things we're trying to feed into this. So to summarize, I guess we're really good at logging, really good at seeing what's happening, like to be better at making that queryable directly. And so, I mean, the reason I'm here and the reason I like doing this stuff is that the goal of it is to make it community developed. And I think you know, we've tried to do a few different things to make that possible. The first is having a fully automated installation, which builds on top of Cloud Bio Linux. The second is to make it deployable in multiple clusters. I mean, it's literally a total showstopper if you go pick up a project and it works on SGE and you have LSF. So you really need a system that works across all those. And so I'm super happy this works across a ton of different systems. It's worked on people's clusters that I never saw, I never logged into, that they managed to set it up and work totally fine on theirs. So it's really cool like that. You can get contributions from people without that sort of barrier straight away. It also provides some. It's not a workflow system. It's a solution that does one thing, but it does provide APIs to sort of tack in new things that if you want it to do. So if you want to add an aligner or variant caller, that sort of stuff is easy. Doesn't make a pipeline for you, but it does do those sort of things. And it's obviously open source, totally hackable. It's got lots of documentation. So definitely check it out on GitHub if you're interested in this. And I think I just want to focus a little on one of the things I think is critical, and I've been doing a lot of work on, is automated installation. I mean, I think the single biggest problem with something like this is that to actually get it running for the first time, there's literally like 20 different tools you have to install, and data files you have to download, and configurations you have to update. And so I've done a lot of work on scripts to bootstrap from like a totally bare machine, boot it up on Amazon, all the way for a ready-to-go pipeline. And the Cloud Bio Linux project is a huge help with this. They already have installations for a ton of these things, so you can build straight off that. And we try to provide example pipelines with real data, so you can get started, you can install it, be sure you've actually installed it, and can run a real pipeline before you start even using it in your tools. So definitely, the documentation has tons more information on that, and the Cloud Bio Linux project is a great piece of infrastructure to use for this sort of work. Finally, I think I just want to talk a little bit about making this accessible to everyone. I mean, I think we're entering a really amazing time in terms of the ability of everyday people, non-scientists, to be able to have access to their genome information, to look at it and learn about it. And I think there's no reason why the tools we develop couldn't be accessible to someone who's interested in using them. This is just a book that came out recently by Joe Dudley and Konrad Karszewski. I always have to be careful when I say Polish names, but about exploring personal genomes. And so it's really sort of telling that you could write a book right now that anyone could buy and pick up and learn how to look at their genome information. And I think you can buy your exome from Baylor for $9,000 if you want to. It's really a cool time, and it's really an opportunity for us as scientists, I think, to make the things that we do available to everyone. And so there's two sort of ways we're trying to do that. The one is Galaxy Project, which I think is more targeted at biologists, people who are familiar with biology, but it's sort of a step in the right direction. So we've been doing a lot of experimental work trying to think of the best way to run a really complex pipeline that has a backend from Galaxy, and so we have custom tools that help build up that configuration file I talked about and run them. And so I think that's a good way to get this available to biologists. I think for the general public, that's a specialized tool, right? So there's a tool from Konrad Karszewski that is called StormSeek that is aimed more at a general everyday person, not an everyday person, I guess, but someone who can figure out their genome and upload it to a website. So it has, this is built on top of Amazon. They literally just have to enter the Amazon information, upload their FASTQ file, BAM file, whatever a provider would give you, and then hit a giant green button called Go. It's very big, so it's easy, right? Because the button is big. I didn't make the good button interface. But this is the sort of tool that we're working on integrating with the backend. So I've been talking to him and set up this uses Star Cluster on the backend. I'd hopefully like to use Polymath and do a ton of work with that. But totally sets up a pipeline that runs automatically on Amazon, spins out all your images, runs it, and then gives you the results back. So in that way, and built with sort of the accessibility analysis stuff we've been working on, you could literally give people the same analysis researchers are using straight away. And you have to be, it's not going to give it to everyone straight away, but I think it's a step in the right direction in terms of making this available to everyday people. And I think that's a goal we should all be sort of striving for if you're working in that area. So in summary, I think that community developed pipelines can overcome a lot of the challenges that face trying to build these complex things. I've been here for a long time, so people know who I am, I guess, but I do it because I believe in community, and I think it's really an opportunity for a lot of people to work on a problem that helps from a lot of people. And the focus of our goal has been, the development pipeline has been assessing the quality. So I think number one, if you're going to have a good pipeline, you have to do good science. Number two is enabling exploration and analysis, so people can take what you've done and easily extend to it. The third is make it scalable, so you can actually do your work and finish in a reasonable amount of time. And then reproducibility, so you know what you did, you debug what you did, and show other people what you did. And finally making it widely accessible to a wide audience. So if you're interested in this, definitely check it out on GitHub. Get in touch with me. I'm happy to help with setting it up, making the installation better, improving it. So, and I'm also happy to take any questions. Thanks, Steve.